After isolation of lysozyme from hen egg white and determining the quality and quantity of the isolation product, the aim of this video is to investigate the enzymatic activity of lysozyme and measure its kinetics using spectrophotometry. First, the spectrophotometer is calibrated by measuring 1 ml of sodium phosphate buffer at 415 nanometers. Afterwards, the bacterial suspension is measured at 450 nanometers. In our case, dry bacteria were used and mixed with phosphate buffer. The value for bacterial suspension should be between 0.5 and 0.75. Otherwise, the suspension should be diluted. In our case, we measured it to be 1.257 and it was diluted 1 to 2. Six large cuvettes are taken and 2.5 milliliters of diluted bacterial suspension was added to each of it. Then 40 microliters of lysozyme as a positive control and each fraction from F0 to F4 prepared in the previous units were added to the cuvettes. The initial absorbance for each is immediately measured after adding the enzyme, and the timer is set up to let it incubate for 5 minutes at room temperature. Don't forget to vortex the sample beforehand. When adding the diluted bacterial solution, don't forget to mix the content up and down with the help of pipette. After 5 minutes, the OD of each sample are measured again and noted down. The contents are then discarded in the table bin and cuvettes are cleaned with distilled water. After determining the presence of lysozyme, we are proceeding with determining the specific activity of isolated lysozyme, which theoretically should be present in F0, F4, and the positive control samples. 2.5 milliliters of fresh bacterial suspension is added to one cuvette after setting the plank again. "100 microliters of first sample F0 is added and immediately measured for OD at time point T0. Every 30 seconds, the sample is measured at 450 nanometers for 5 minutes. The same procedure is repeated for sample F4. In the end, the procedure is repeated again for lysozyme positive control.
The experiment is now done, but how can you get the specific activity? We will work in Excel to figure this out. Here you see the table where we noted down our values during the experiment. First, I transferred the values you just saw into a table in Excel, and then you have to create a plot where you plot absorbance versus time. So you get to insert the graph, then you make a right click and get to insert data. Then you click on insert, you name your data, then give the x and y values of the plot. Then you click on insert another, also name this one, click on x values and again choose the time points. And again one last time for the lysozyme. And again choose the time points, and the y values are the lysozyme absorbance values. Then you press on OK, and you get this wonderful graph of absorbance versus time. With this plus, you can add title or axis titles. This is what I did here, and also the legend. And then I named the axis and also the diagram. When you scroll down, you see I already prepared kind of a table. You start by calculating the total absorption reduction, where you subtract the last OD value from the first. Mine were in the E column, so I chose them. Then you divide this value by 5, as we measured in 5 minutes, but we want the absorption reduction per minute. This value you have to multiply by 1000 in order to get the units in the sample. For the units per milliliter, I prepared kind of a help. So 100 microliters times 10 equals to 1 milliliter. And if we have 7.8 in the 100 microliters, we also have to multiply then with 10 in order to get the units in the sample per milliliter, which equals then 78 for us. After that, you calculate in the same way the total absorption reduction, the absorption reduction per minute, the units in sample and the units per milliliter for F4 and lysozyme. When you scroll down a bit again, you see I prepared again kind of a table. There I inserted our values from unit 5 for the total protein concentration of each of the 3 samples. Because for the specific activity we have to divide the units per milliliter by the total concentration we measured in unit 5. So what you can see here is I divide the value we have at the end, units per milliliter, by the concentration we measured in unit 5. And this is the specific activity, which you now figured out for lysozyme. And yes, that's it. This is not only the end for this tutorial, but also our last video of our lab tutorials dealing with lysozyme. We had a lot of fun in the lab and hope we could help you a bit with these tutorials. Have a good one, everyone. Bye!